Today it is our great pleasure to host our absolutely outstanding colleague, Don Cleveland, who has an amazing history, as you all know, but some pieces that you may not know, and I didn't know until I looked at his CV, just as a refresher. He was born in Waynesville, Missouri, which is an hour and 20 minutes from the place where I went to high school and college, Springfield, Missouri. Then he's very proud as a graduate of New Mexico State. And I went to Southwest Missouri State, which is even less. It's got two directions in it. So, but Don after that went to Princeton, which is where he really, I think, started establishing incredible work. And then I don't know, after that joined us. Oh, went to UCSF and all that, but then eventually came to us. But as you all know, he's incredible impacts. Discovered tau. I mean, that's amazing. It really brings great accolades to this center. And Don's been doing incredible work in Huntington's, Parkinson's, ALS. I mean, you name it. He's made incredible impacts and has won the Breakthrough Prize for his efforts in ASOs, and we're going to hear about that today. So, ladies and gentlemen, the one, the only, I bring you Don Cleveland. Hello, Cleveland.
Wow. I think we all realize that truly was over the top, but thanks, thanks to you for the opportunity to talk about what we've done. And I've prepared this in a three-part story about the development of antisense oligonucleotides and their use in treating human neurodegenerative diseases. That work, as I'll show you, that initiated completely here at UCSD. Tim Miller and Richard Smith, along with my colony manager, Melissa Macalonis, they did the founding experiments that, and I'll show you how we got there and then where we're going. And of course, this community, you all know that where genetics has implicated a gene or genes as causative or at least contributory to the major diseases of the aging nervous system, Alzheimer's, Parkinson's, ALS, Huntington's, all of those genes are widely expressed. None of them are uniquely expressed in individual classes of neurons whose failure, degeneration, and death gives rise to the disease phenotype. And indeed, again, I suspect this community all knows this, that it was the discovery of Andy Singleton and his colleagues at NIH originally that if you inherited 3 copies instead of 2, 3 copies of a completely wild type alpha-synuclein gene, you will develop Parkinson's disease. And that was followed by the discovery that if you inherit 3 copies instead of 2 of amyloid precursor protein gene, no mutations, just a wild type gene, you will develop Alzheimer's disease. And so that was clear that a therapy in those cases would be just to lower the synthesis of the overexpressed gene product. And so that's what we thought. And the beginning in 2003, 2004, can we just turn off a gene? And if you had an audience of young molecular biologists, they would all say, oh yeah, how are you going to do it? And the answer is you'd use an siRNA, or today you'd use a CRISPR, but both of those are still yet to be demonstrated to be actually useful in human neurodegenerative disease. And the approach we chose was the older approach, a single-stranded DNA. If you can get it into the target cell cytoplasm, so it can diffuse into the nucleus, that little DNA, single-stranded DNA, can pair with a target RNA, even a pre-mRNA, and this RNA-DNA hybrid is then recognized by an endogenous intranuclear enzyme RNase H, which cleaves the RNA but not the DNA. And so this is an RNA catalytic destruction mechanism, which relies upon the presence of an endogenous enzyme, which is present in the nuclei of almost all of our cells. And over the years, the medicinal chemists have modified these DNAs to thiophosphates instead of, and to the changes to the 2' or 5' position of the ribose sugar. And those changes, the current changes, have given these designer DNA drugs have given them remarkable properties. So I'll show you in a moment. But one of the features that, at least for the current generation of these antisense oligonucleotides or ASOs, is that if you introduce them into the periphery in any manner, there's zero that gets into the nervous system. I mean zero. So in the examples I'm going to show you today, in the human examples, it's intrathecal injection into cerebrospinal fluid, and in the rodents, it's an intraventricular administration. And when we started this, there were some problems, like everybody knew it couldn't work. And I mean, it's absolutely true. Everybody knew it couldn't work. And these are 10 to 15 times typical drug is 500 Daltons. These are 7,500 Daltons. That we're using 18 or 20 MERS. They're heavily negatively charged. How would you be able to deliver them broadly throughout the nervous system? All the textbooks said this can't work. And actually the beginning of the breakthrough was we did it anyway. And we did it, for some of you, Richard Smith, local neurologist, and he was adamant that we try it. And if you know Richard, he was adamant. And we did ultimately try it because he just absolutely insisted. And he was so annoying. I thought it'll just be easier to do the experiment than to have to put up with that again. Okay, so and we now know, and even in the founding work, we had demonstrated that if you do infuse these, directly inject them into cerebrospinal fluid, that you can deliver them almost everywhere in the rodent. Oh, but even more so, almost everywhere in the non-human primate. And here we showed that in the founding work, and then this one is taken from a recent publication of Ionis. Oh, I forgot to mention, sorry, important omission. On slide 1, I am a consultant for Ionis, a local biotech, and I have been a consultant for them since 2003 or 4. And here is the ability to get target RNA degradation in the vertical axis versus part of the nervous system in a non-human primate dosed intrathecally, and you can see, man, you can really target dose in a dose and time dependent manner. You can lower a target gene in the spinal cord, in the cortex. Most brain regions, not quite all, but most brain regions you can reach with this kind of approach, even in the non-human primate. And the disease we started with was ALS, selective degeneration and death of upper and lower motor neurons. The paths of those neurons, the upper motor neurons sending their axons down the spinal cord and synapsing either directly or indirectly onto the lower motor neurons, whose job it is to trigger muscle contraction, as drawn here by Cajal. And the beginning of the molecular era of discovery in ALS was the identification that about 10% of instances are clearly inherited and through dominant mutation. And the founding paper was this one, Bob Brown and colleagues worldwide, identifying the gene mutations in the gene encoding cytoplasmic superoxide dismutase is causative of ALS, about 2% of overall ALS, about 20% of inherited ALS. And the original proposal in the News and Views article accompanying this report was that these mutations were loss of activity. And they were point mutations. There were 11 of them, and they were all point mutations. But it was declared that it was loss of activity because everyone knew that oxidative damage drives neurodegenerative disease. And here, SOD1 is a homodimer, and there are now more than 140 different mutations known. Almost none of them preclude the synthesis of a nearly full-length polypeptide. And just the genetics alone really called into question the conclusion that it was loss of function. But we tested that by making, of the original 11 mutations, we made 6. And I chose them. They were all the charge shifts. And here, glycine to arginine at position 85. And we showed that this one has 0 activity as a dismutase. Another one has 100% activity as a dismutase. And we made transgenic mice with both of those, the 2 outliers. And the answer is both of them cause fatal heritable paralytic disease. And this one, where the enzyme itself, the mutant enzyme, has zero activity by any assay, then a postdoc fellow with me, Lucy Bruin, she took those. It has a transgene, an inactive transgene, and 2 endogenous alleles. So that animal had 100% normal dismutase activity, but then she tested, is it true that dismutase activity plays any role in what? And so she deleted one of the endogenous genes, or both of them. And as you can see, that has no effect on the disease course, on any aspect of disease course. So this led to, after a contentious period, it led to a consensus view now that these mutations provoke disease through acquisition of one or more toxic properties, not through loss of enzymatic activity. Oh, that's a little sobering that in 2024, now more than 30 years later, we don't agree as a community on what the toxicity is. I think there are 9 models, and I think everybody's probably partly right. And then the question is, where's the toxicity? And so this is ubiquitously expressed gene product. And so is it really true synthesis within individual motor neurons is what drives the degeneration of those neurons? And the initial experiment, we made chimeric mice, mixtures of normal cells and mutant expressing cells at very high levels. And here, in this particular example, we marked the axons cleverly so that they were, the red ones were wild type and the green ones were mutant expressing, because we had antibodies that would bind to the human mutant protein. And here in multiple animals, even with 35% of total motor neurons mutant expressing at high levels, none are lost. There's no degeneration. It's not from individual synthesis within individual neurons. And indeed, we've further then built a transgene encoding the human mutant protein, which was flanked by lots of recombination sites so that the action of Cree would excise those genes. And so that's now become a widely used tool, but this was the first one used in the neurodegenerative disease context, where we now could then selectively delete mutant synthesis in individual cell types. And when we did that, we discovered that it absolutely, synthesis within the target motor neuron of the mutant SOD1 does drive early disease onset. But if you, in the surprising findings, is that if you remove the mutant gene product from microglia, the innate immune cells of the spinal cord, you have no effect on the timing of disease onset, but you triple survival after onset, something which, of course, if that could be achieved in human practice, would be seen in the New York Times as a cure for the disease. And indeed, that was the founding one. And then the next one, we removed it from the astrocytes, and that too had no effect on disease onset, but doubled survival after onset. So the glial cells, synthesis of mutant SOD1, at least in these rodent examples, are driving rapid disease progression. Now, okay, so, and then we looked at many other cell types as well. So, and when we proposed that the disease process was therefore non-cell autonomous, not from an action of the mutant protein within an individual neuron, that was met with hostility. And it reminded me of Schopenhauer, a German philosopher in the late 1800s, who said, new truth passes through 3 stages. And first, it's ridiculed. Yep, that happened. Then the second phase, which I've seen repeatedly in my career, it's a little counterintuitive, but then the hostility gets even stronger. And then third, it's recognized that it was self-evident and that it was always known to be true. And that is exactly what went there. And even in the AD world, there was a very neurocentric view of AD for a very long time until the TREM2 was finally recognized, and that actually maybe the non-neuronal cells might be doing something important. And I'm quite confident that we're almost at phase 3, which is because it is absolutely obvious. But okay, so all right, so here's the founding experiment done in the Palati vivarium of injecting an ASO to suppress an ALS causing mutation in SOD1. This is in a transgenic rat, which has a very aggressive disease course from once the disease starts. And here you can see near this, and we're injecting into the ventricle. So near the site of injection, we can lower in a dose and time dependent manner, lower the target RNA as far back almost as we can, in this example, we can lower it to as low as we want. And oh, a little farther away, oh, look, we can lower it throughout the entirety of the nervous system of this animal. And if we dose them at disease onset. Well, I said it was a hyper-aggressive model. So mock-treated animals only live about 25 days after disease onset before you have to sacrifice them for humane reasons. But we doubled survival after onset by dosing at onset in a founding experiment, which again, if we could do that, oh, we maybe have done that. If you could do that in a human context, then that would be clearly seen as a real disease-modifying therapy. And indeed, that the phase 3 trial of using an ASO in SOD1 mediated ALS was reported here now 2 years ago. And here was in the first 6 months, the green curve, those are the treated patients, the dashed curve are the placebo patients. And they in a 6-month trial, the trial with the number of individuals in the trial did not meet the hoped for statistical measure, but in the open label extension, in a trial that was co-run by Tim Miller, who was here as a in neurosciences. He's an MD, PhD neurologist. If you follow those in the Washington University cohort of patients, 25% of those patients have improved in almost every muscle measure 1 year after therapy. They're getting better, and so the trial was just too short, and it failed in part because the placebo patients did way better in the phase 3 than they did in the phase 2 because they were earlier in disease. And so it was it was either a statistician's error, anyway, it's a trial. The trial was not designed optimally, but indeed, it's now been approved in the US and in Europe. Okay, so now, and Jim mentioned in the introduction, yeah, so there are multiple places where you could use a gene modifying therapy. And indeed, as probably all of you know, the most frequent genetic cause of Parkinson's disease is a mutation in the LRRK2 kinase. The mutations are not loss of function mutations. They're hyperactivation, so that there's too much kinase activity. So it would be an example of where if you could use a conventional drug if you had a selective inhibitor, or you could use an antisense oligonucleotide just to turn down synthesis of the LRRK2 gene. And it was very kind of Jim to mention that I did get the breakthrough prize, and it was handed to me by Sergey Brin, the founder of Google, and is very public about it. But I did not know at the time he handed it to me that he is a LRRK2 carrier. His mother is a homozygote for the LRRK2 mutation, and so he's at high risk of Parkinson's disease. And indeed, we have gone to trial with Ionis and Biogen, gone to trial to use an ASO to suppress LRRK2. And indeed, back to where I started, which was that triploidy for alpha-synuclein is causative of inherited Parkinson's. And so, and almost all Parkinson's patients accumulate alpha-synuclein deposits. And so maybe turning down the alpha-synuclein gene will be a non-disease mechanism therapy. And indeed, that in 2020, the phase 1, phase 2 trial was initiated to suppress alpha-synuclein with an ASO, not quite in Parkinson's disease, but in related disease, multiple system atrophy, which is a genetic cause of which is mutations in alpha-synuclein. So Biogen's just trying to double down on the likelihood of success. Okay, and then One additional example about use of an ASO. Jim did mention this. Yes, this is a young graduate student who purified tau. We're now in the 46th anniversary after that. And we did show that tau was heterogeneous and that it was a natively unfolded protein. We showed that then, we just didn't coin the phrase natively unfolded. And this community will now certainly recognize that tau misaccumulates and tangles inside the affected neurons in Alzheimer's disease and in chronic brain injury, and that most people agree that the tangles of tau are the best correlate of cognitive decline. They're certainly highly correlated with cognitive decline. And indeed, there was a long discussion when I tried to purify tau in the first place, because here was the purified tau. And you could see it's not not a single species on an SDS gel. And there was a long discussion, will he ever graduate? Since he cannot purify it. And what I did, we invented in a method, and here we cut these bands off an initial gel. There they are, cut one. And now you can see I cut the second one is actually, you can see they're actually doublet, 2, 3, 4, 5, 6. There are 6 tau, we saw 6 tau isoforms. And when you peptide map them, they give almost identical peptide maps with either of 2 different proteases. And so it's like, oh, they're very similar. And I showed that actually each one of these species was multiply phosphorylated. Using 2D gels, you could do that. Mass spectrometry, you cannot do that. And I showed that they had up to 5 phosphates on each one of them. So there were 30 tau species that we identified there, but they were largely the same. And here, now that this is an educational institution, so I thought I'd bring a quiz. So these 6 tau isoforms that were present in the original preparation, we did not propose that they were the targets, that they were the products of a single gene with alternative splicing. And so why didn't we propose that? That's the quiz. Right. So yeah. So if you have this is really good when you have, and I apologize to the audience already, but if we had a younger audience, they have a great deal of trouble with this because yes, splicing was discovered the next year. So yeah. All right. So, but just to make sure everybody saw it, that here a year and a half ago, the outcome of a phase 1 trial to suppress tau in AD or in mild cognitive impairment. And here, the data from that trial, yes, and you could suppress tau in a dose-dependent manner. And you could total tau or phospho-tau, both of them. And even more so, Biogen released the imaging. And so in particular, there were 2 individuals who in the first 6 months were in the placebo arm of the trial. Here they are. And here are the tau deposits imaged with PET imaging, the tau deposits seen at trial entry. 6 months later in the placebo arm with apparently larger deposits on, like here on the left-hand side or here on the right-hand side. And so these individuals were actively depositing tau. And so were in active disease, and then they were converted to drug arm. And then because COVID disrupted some imaging points, but a year later, they both were imaged again, and what you can see is the original tau deposits were largely disappeared. So and that was true in 11 of the 12 patients for which had been enrolled in this imaging strategy. So I could hardly have come out as one of those who's a cheerleader for tau thing and cheerleader for ASOs. This could hardly have been better, except Biogen released no information at all about cognition. They have now announced that they're going to publish those data in the coming year. Yeah. So these ASOs, yes, they're generic tau. Although Tim Miller did all of the work the initiating work for this. He showed that he could use splice altering ASOs as well. He demonstrated, I think, what our community thought broadly, that 4 repeat tau was more, 4 microtubule binding repeat tau was the bad tau. He demonstrated that in mice by taking mice that expressed human tau, mostly 3 repeat tau. That was disappointing. But then he converted it to 4 repeat tau at exactly the same gene, and it made it much worse. So it showed that 4 repeat tau was clearly more toxic, at least in the context of the rodent examples. But here, yeah, this is suppressing total tau. And of course, if you're going to suppress tau, you might think about National Football League disease. The founding one, the founding individual, Mike Webster, tough guy, defensive player, and of course the most famous San Diegan, Junior Seau, both dying from this chronic traumatic brain injury, and the disease is diagnosed post-mortem by deposits of tau. Led by work by Ann McKee at Boston University, and here deposit of tau, focal deposits, and if you blow one up, it kind of looks like there's focal damage with maybe a slowly spreading tau. But as long as if tau is really more than a marker of damage, and if it is a part of the damage, then suppressing tau would be a viable approach. And the phase 1 trial just been completed, so we should be dosing our football players with. Yeah, I say that with a smile, but actually also with maybe true, if it's really true that tau is playing a causative role. And then one last example of this. So what are we going to do about TDP-43 pathologies? And so it was discovered by Virginia Lee and John Trojanowski in 2006 that an RNA binding protein, TDP-43, typically is supposed to be mostly intranuclear, but in disease examples, it clears from the nucleus and many times deposits in the cytoplasm. That was recognized initially in ALS, where almost all examples, except the SOD1 example that I started with, have this TDP-43 loss of function and aggregation in the cytoplasm, and about half of frontotemporal dementia has that too. And more broadly, as people have looked more and more, my view of the current AD literature is there's an agreement that at least half of the AD patients have TDP-43 abnormalities. All of in a disease called late, and the oldest individuals, 100% of those have TDP-43 pathology, and we're being referred to as TDP-43 proteinopathies, and so it's broadly true in neurodegenerative disease. And while we showed originally that there were modest changes in 1,500 RNAs when you lowered a TDP-43 within the rodent nervous system, 1,500 RNAs changed in abundance or in splicing pattern, we missed all of the interesting ones, because they're not the most affected ones in human neurons are not affected in mice. And the one in particular, here we've used an siRNA to suppress TDP-43 to 25% of its original level. And there's an abundant RNA which is suppressed even more. And it encodes a protein called Stathmin-2. And in John Rabbit's laser capture, is laser captured motor neurons, in the surviving motor neurons from individuals who died from sporadic ALS. When he did capture those and then the modern RNA sequencing experiments, it's the 20th most abundant RNA in human motor neurons. And we showed that it's required for regeneration after initial injury in cultured motor neurons. And we just demonstrated that in mice as well. So it plays some role in stabilization, and we know it plays a role in structuring axoplasm. And I took all those slides out today, but we know a lot about what it does. And so we argue, why is it lost in a TDP-43 dependent manner? And here it is, we suppress TDP-43 to about 25% of its original level. Ooh, and look, at the protein level, stathmin disappears. It is the most sensitive. There's universal, almost universal agreement that stathmin is the most sensitive. The RNA for stathmin is the most sensitive. And we know why, because it gets cryptically spliced. Here is the structure of the gene. Intron 1, there's a cryptic 3' splice site that gets used when TDP-43 is depleted, and that's followed by a cryptic polyadenylation site right there in what should have been intron 1, making a completely non-functional RNA that can't, that absolutely does not encode stathmin. And we know that's true in human patients, again, from John Rabbit's laser-captured motor neurons in normal individuals. If you do an RNA-seq experiment, you see exon 5, exon 4, exon 3, exon 2, a 3' bias to an RNA-seq experiment, and here from 4 different individuals. But if you look at those who've died from sporadic ALS and you recover motor neurons, the surviving motor neurons from them, you can see that the protein coding exons are nearly missing because of cryptic splicing and polyadenylation of stathmin-2, almost complete loss of stathmin-2. And if you do that in rodents, a 1-year-old mouse, and suppress stathmin chronically, the axons collapse, and you drive denervation. Within 8 months, you drive denervation of 2/3 of the motor axons, the initial steps of ALS. And so we argue, and we know how TDP-43 functions. And here I just mentioned it, but if you suppress transiently with an ASO, you drive denervation by just suppressing stathmin. And if you do it chronically with an AAV encoding an shRNA, you really get down to 2/3 of within 8 months, 2/3 of the neuromuscular junctions are denervated. So you can drive ALS by suppressing stathmin. And we argue from that why don't we then use an ASO which has been modified so that when it pairs with a target RNA, the hybrid, DNA RNA hybrid, is not recognized by RNase H. It is not going to be cleaved, but it's going to be bound by the oligonucleotide and thereby try to suppress the use of the cryptic splice site and the cryptic polyadenylation site. We did many molecular biological experiments. My graduate student, Michael Bond, did them. And we know that it's actually the cryptic splice site that drives everything. If you remove the cryptic splice site, the cryptic polyadenylation site is not used. It's all driven by cryptic splicing. So here we tiled across this array of intron 1 in cells, in neuron-like cells that have TDP-43 mutations that lead to partial loss of function. And we ask, can we suppress the cryptic splicing? And the answer is yes, in a dose-dependent manner for the blue ASO, yes for the red ASO, here they are. And even more importantly, can we restore the full-length gene product? Is it now being processed correctly? And the answer is yes, completely correctly. It's almost complete correction with either the blue or yellow or red ASO. So we argue from that actually, the discovery of stathmin as a target was a pair of papers from our team, the UCSD team, and a team of Kevin Eggins then at Harvard. They were published back-to-back in 2019. We demonstrated last year by humanizing mice so they now have a human stathmin gene, or humanized for the misprocessed region, that indeed we can, in the mammalian nervous system, we can restore correct splicing even without even when TDP-43 is not bound to the RNA. And we expect with Ionis to get to trial in 2025. A small startup company from Kevin Eggin actually went to trial without evidence that their ASO is effective in the actual nervous system. It's a dangerous approach, but they may be successful. I wish them success, but Ionis would never have gone forward without demonstrating efficacy in an actual animal system. Many ASOs that work in cell culture do not work in the nervous system. Human cell culture, yeah. So they yeah.
If the whole thing gets spliced out as an intron.
they can show that they can restore. They showed that in an iPS-derived motor neuron that they could restore synthesis. But we know, working with Ionis, that it's just it's not explained why ASOs that can be very effective in cell culture, many of them are really not very effective, either from distribution or uptake. I don't know. If Ionis knows, they haven't shared that with me. So, but many don't work. I hope they, yeah.
I mean, if it's the animal model.
Yeah, yes. Of course, the animal model isn't perfect, but at least if you humanize, again, they didn't have a humanized mouse to be able to test in, and so we humanized the Staffman allele, and we could then demonstrate that. And here's the scorecard from where we are today. We know that we can use these ASOs broadly in rodents and non-human primates. We can use them for catalytic RNA degradation or altering splicing. And what I didn't mention, I forgot to mention, key part, they're the best ones. They're long lasting, so that in single dose injections, it gives you 3 to 4 months worth of efficacy in the nervous system. They're certainly commercially feasible today, and there are multiple examples of them being safe. The founding example of approval was in spinal muscular atrophy using a splice correcting antisense oligonucleotide work from Frank Bennett and Adrian Craner. Frank Bennett's a CSO of Ionis, and Adrian Craner at Cold Harbor. This is kind of spectacular. There's kids that typically you guys know, that neurologists know, that it's the typical disease survival of a year with really very paralyzed, and now the oldest ones dosed early enough in disease are 7 or 8 years old and they're walking. It's kind of a miracle. And here we know it's been approved for SOD1 mediated ALS. There are multiple ongoing trials. There were failures in the most frequent cause of ALS, which is expansion in the gene, hexanucleotide expansion in gene C9orf72. That failed. We're confused. The initial trial in Huntington failed, but Roche is in conducting a second phase 3 trial with 350 patients, insisting that the patients be much earlier in disease than they were in the first trial. And I talked about the trial in AD to lower tau, or in the 2 trials in Parkinson's to lower LRRK2 or alpha-synuclein. And now what I'd like to do is to finish with one final example of what you could do with an antisense oligonucleotide. And a group like this will remember that the great Ramon y Cajal had recognized now 100 years ago that there just aren't many much adult neurogenesis, and he added that it's the duty of the next generation, and maybe I should put an S on there, next generations, but to find a way to overcome this intrinsic failure of the brain to regenerate. And so what I'd like to close with is the beginning chapter of trying to really do that. So can we use these antisense oligonucleotides to reset cell identity? And indeed, or can we get them to reactivate dormant neurogenic niches to produce new neurons as a means to replace those lost to disease? And the strategy began when by one of our former colleagues, Zhidang Fu, who was here. He's now at Westlake University in China. But it was his strategy based on molecular biological experiments with cells in dishes, was that there was a 3-gene circuit. There were 2 3-gene circuits. The first one for establishing neuronal identity. And the first one is here, with 3 components, each of whom which inhibit each other. And if that were true, then the circuit will be frozen in a position if you have a high level of any one of these inhibitors, because they all inhibit each other. And one of them is PTBP1. It's an RNA binding protein and functions in one or more aspects of RNA maturation. And in the neuron, it has little PTBP1 and whereas glia and neuronal precursor cells have higher levels. And so maybe if you could just suppress PTBP1, you would get glia to convert, or a dormant precursor to turn back on. And so the strategy that we're going to use is with an assistant professor in cellular molecular medicine, Bogdan Bentu. He was trained with Zhao Zhang at Harvard, the developer of one of maybe the most popular spatial transcriptomics approach called MERFISH. And what we've done to it is to add to it the ability, and we're calling it multimodal MERFISH, because in a tissue section, we can measure the transcriptomes of 80,000 cells in the intact tissue, and we can use immunocytochemistry to find up to 10 different proteins. And we can find, if you had, for example, in Huntington's disease, you had somatic expansion of a repeat, we can find the repeats, we can measure the lengths of the repeats in all of those 80,000 cells. Okay, so, and one of the beauties, or the way this really works, is you take your library of probes, and we're using 48 probes per RNA. The examples I'm going to show you, we're doing 1,200 genes, so that's 1,200 genes times 48 probes, lots of probes. And we're going to hybridize them all together in a tissue slice. And now we're going to polymerize a thin acrylamide layer, and each of those probes has an acridite linker, so it will be covalently frozen in its position. So we're going to hybridize it to the original target RNAs, this complex library of thousands and thousands of probes. And we don't care now, because the probes are now frozen by the acrylamide gel. We don't care if the RNA gets degraded after, because we're only reading out the probes that were frozen at the beginning. And there, in cycles, I'm sure all of you have seen these kinds of UMAP projections of purported cell types. You think, okay, but the thing that the spatial transcriptomics gives you is you can now take each of these clusters, we've numbered them here, and you can ask, where are they? And you can see by going through that, that at least for many of them, there really are cell types there. And if you look at the kind of cell, you can identify them from the transcriptome. And so in the founding example, there were 22 kinds of neurons that were identified, and 8 kinds of astrocytes. Remember Ben Barres trying to find A1 and A2 after that heroic thing? You find 8, and there are 7 of which are very focally localized, and the 8th is everywhere. It's probably a precursor. Okay, so, and what can you learn from that? So if you do that, so you follow those genes, so 1,200 genes, you in an 8-week-old mouse, and you now find the clusters. Here they are. These are clusters of cells. There are 3 kinds of cells: ependymal cells. There are these yellow cells, which are neuronal progenitors. They could be progenitors, and if you know the Albert Bourla, he calls them type B cells. And here are the immature neurons. And so we can see them by here. These are the UMAP clusters. And we know exactly, and you're going to see in a moment, where they are. We know exactly where they are. But if you look at a 1-year-old mouse, and you probably have heard that Rusty Gage has worked very hard on adult neurogenesis. But adult neurogenesis is in very young mice. So, because in a 1-year-old mouse, there are almost none, not quite none, but almost none. There are the precursor cells, but there are no newly made immature neurons. And here you can see that in the young mouse, we can see that the precursor neurons, the yellow ones, they're lining one wall of the ventricle, and then they're streaming out from the ventricle, heading toward parts unknown, but including the striatum. But they're nearly missing. The precursor cells are still there, but they just don't have any of these pink cells. Now, what happens if you transiently suppress PTBP1? And the answer is, you get them back. There's the new, we have taken the dormant precursors and they have now restarted and now are becoming neurons. And even more so, we were really worried that we would just deplete a dormant precursor pool, and that's all you could do. But there's even more yellow cells after treating with PTBP1, and we used a DNA analog to determine that they have undergone DNA replication, probably 2 rounds of DNA replication. So we are left with the model not established, but the model would be, we've reactivated them to undergo 2 rounds of division. The last round of division probably is an asymmetric division where one of the 2 became a neuron and the one stayed the immature one. In any case, we have more. And so what we propose is that we can make even more by doing it again and then doing it again. That has not been attempted, much less achieved today, but it certainly will be. And you can see they're beginning to stream out. There were also a handful in the dentate gyrus, and we had shown, I didn't focus on those today. We know a lot about that. You can make new neurons in the dentate gyrus by transient suppression of PTBP1. Okay, so we're arguing that we can use these antisense oligonucleotides to reactivate neurogenesis in the aged mammalian nervous system. We're identifying that by using this multimodal MERFISH technology. With Bogdan, I've invested very heavily in that technology. It has way more power than the commercial systems. These are home-built microscopes. There are 4 of them now. They're assembled from 200 parts. They're assembled by Bentu by 200 parts.
Once you've kind of demonstrated that your spatial, once you've demonstrated that your transcript maps onto the populations in their spatial transcript,
yeah,
probably go back and simulate that move.
You could, but there's actually there's no need to do that actually, because the real issue, Jim, is that if you hear people talk about single nuclear RNA-seq, single cell transcriptomics, the read depth is so shallow that it's not true that they get individual cell transcriptomes. What they do is they get UMAP spots, clusters, and then you draw a circle around that cluster and you add up all of the reads within that. And we did that, for example, I should have put that slide in here today. If you use single nuclear RNA-seq and you say, for say a motor neuron, and you say, well, what marker genes do we use for motor neurons? And we, the big W, we typically use CHAT, chloramphenyl acetyltransferase, and you need it to make acetylcholine. And if you use our approach and you say, how many RNAs are there for CHAT in a human motor neuron? 600, 10% of which are in the nuclei. If you do nuclear RNA-seq, how many do you get? 1. So the most frequent number is 0. And well, there are two competing single nuclear RNA-seq approaches. The one gets 1, and the other gets 0.01. So the read depth is so shallow, even for the marker genes, you can't get them on an individualized cell basis, but we can. And the beauty is to take the nuclear RNA-seq where you're using pooled similar like cells to identify all the genes that you really want, all the genes that are really expressed, and then make a library of genes with these probes that includes all the expressed genes in each of the cell types that you care about. And if you do that, and you have a 2,000 gene set, you absolutely can get the complete transcriptomes of all 80,000 cells on a single cell basis without assuming that they're all kind of similar. Doug. Can we make probes? Well, we haven't, of course you can in the examples I showed you before, but we're in these readouts. So the question, I think maybe I misunderstood your question. It was that, can we actually identify RNAs that are misspliced? So the answer is sometimes. It depends on really whether those RNAs, if there are stable enough RNAs from the misspliced part. There are many that are substrates for nonsense-mediated RNA decay, and those you probably can't find. But Stathman, yes, you could find, and you would have new sequence, which was now in the Stathman case, from intronic sequence, which would now be in the cytoplasm as a stable RNA, and you could find it. Yeah. So sometimes.
I'm thinking we in the ADRC on the announce afterwards, like out of ranks and things, and this is very exciting as a tool to potentially enhance the impact of some of the science. But I'm sure this tool is pretty overburdened.
Yeah. So
maybe you could still expand your local impact by saying, okay, can't you do your whole experiment on it, but you can validate your
correct. Yeah. Yeah. So it's in its current example, it's in my view, it's extraordinarily powerful, and it has extraordinarily low throughput.
Yes, that's right.
So that's we're oh no, it's all here. No, it's in CMM East. No, there and Bento is assembling quite a number several of these. I've paid for 2 of them. But the throughput is the key part. But there, the question is, how many genes do you actually want to look at? So it can be, it's very slow data collection. If you want to do, we're planning a version 2.0 library. You buy it from a company called Twist. They make 100,000 probes for you for about $20,000, and that's an infinite supply. You'll never use it up. You will just be worried about version 2.1 or 2.2 or version 3.0. But you don't have to read them all out. It's actually, the readout is combinatorial and complex. We have only 3 colors, and cytoplasm is crowded with RNAs. So you can't try to read all of them out at one time, because you'll have them superimposed in loci, and so you won't have them uniquely identified. So there are issues, but the idea that you can really do it on a single cell basis, absolutely you can do that. And so yes, you could use it to validate ideas that were proposed otherwise. But what's really good is to take the single nuclear RNA-seq to educate your libraries, and then use that. And so we're just about to pay the $20K for version 2.0 of our library, and then. But you can read out 200 genes very fast, or you can read out all 2,400 genes very slow. Yeah. Okay, so I almost finished. So let me just say one last thing, is that we do know that in the human example, we've only, that if you look at ventricle kind of collapsed in this particular example, but the progenitor-like neurons are present, just as we had said in the mice. They are present. They're in exactly the same place. They're the dormant what have the transcriptional hallmarks of dormant precursors. There they are, the yellow cells, just where they were in the mouse. So we argue from this that it's really is possible that we'll be able to reactivate neurogenesis using these sorts of approaches in the primate as well. And who's we? And so in the 3-part story I showed you today, the last part in using this answering Cahal's challenge to make new neurons, it was really led by Roy Mamon, a current postdoc, and Sonia Vasquez Sanchez, who I have to say, she is the one who made this a truly powerful approach, along with Bogdan. Without them, it absolutely wouldn't have happened. She identified conditions where all of those multimodal things can be done on exactly the same cells. And then the Stathman team of identifying a target for TDP-43 proteinopathies. It was 2 postdocs, Yoni Lopez-Orozcan, who's now a project scientist with me, Zeev Melamed, who now at Hebrew University, 2 former graduate students, Michael Bond, molecular biology whiz, My partners in this, Clotel Nagier-Turian, now at MGH, there was a partnership as well with Jax and Kat Lutz, who built these humanized mice that I described briefly. And I didn't mention it yet, and Neil Schneider at Columbia has really showed 2023 was a watershed year in ALS because there were two examples, the first approval of the Ionis drug for SOD1 mediated ALS, where we now see partial disease reversal after 1 year of dosing. And Neil Schneider has shown that for a second example, mutation in the RNA binding protein FUS, which is also causative of ALS. And you can argue it's on an anecdotal kind of basis, but treated patients have had remarkable disease reversal. We know that this disease, 2023, ALS is a treatable disease. And then lastly, the use of these designer DNA drugs, it was really, I mentioned Richard Smith, the local neurologist, was adamant that we try it. Tim Miller, then a fellow with me, now co-ran the trial with Merit Sukovitz and demonstrated that using ASOs against tau are really, really helpful. Melissa McAlonis, who did much of the original surgical manipulations, and Frank Bennett, the CSO of Ionis, and our approval last year. Thanks very much.
All right.
50 people online.
Super. So I wanted to say something that just, first of all, really thank you. It's just awesome to see the kind of work going on. Always something new, always something incredible. The other thing I failed to mention at the beginning was how many of those young scientists Don has mentored. Basically countless junior faculty and graduate students and postdocs. And it's something that the ADRC would love to fuel across the entire community. A lot of those individuals did get some either award through our T32 grant, our Alzheimer's focused T32 that Eddie Koo and now Doug and Shu Chen are running. Please keep your eyes out for that. But one that is hot off the presses is our Rec Scholar program, which comes with money, and our developmental project awards, which comes with money as well. So we would love to have you guys keep everybody online, everybody here, keep your eyes open for those announcements, because it's one of the things that we at the ADRC are take a lot of pride in through your mentorship and the other leaders in the field here at UCSD. So Don has built unbelievable pipeline of incredible talent that we saw, and I take a little pride because some of those people were people we funded a little bit.
Yeah.
And through his leadership of CMM, also at the junior faculty level, building such an incredible team. So thanks, Don.